This is Views and News with Clarence Ford.
Only on Cape Talk.
The Naked Scientist, Dr. Chris Smith, joining us from the UK, a consultant virologist at Addenbrooke's Hospital in Cambridge. Dr. Chris Smith, also, of course, known as the Naked Scientist. Welcome, Chris. Great to have you with us.
Hi, Clarence. Been thinking of you a lot this week because, of course, South Africa finds itself at the heart of an international health situation with the business with hantavirus, which is dominating headlines. So maybe we could begin there.
I think so. And I think we are, with COVID, a recent past, we know of 4 infections. That's in the news today. And I was feeling the trepidation. I was getting a bit of that post-traumatic stress just hearing that there are 4 infections in Cape Town.
Well, it's sort of having, I think, repercussions and giving people those sort of twinges of what happened during the pandemic 6 years ago, because of course that began with, or right at the beginning of that, were lots of people stuck aboard a boat. You remember the Diamond Princess? I think that was over in the Far East, though. Lots of people couldn't get off because no one knew how to handle cases of COVID. And so this kind of had some echoes of that. Little bit different. We're dealing with a very different virus, a different entity that's very different in its infectivity and its ability to spread. But it's nevertheless interesting in the respect that it points to and reminds us that emerging infections, which this is an example of that, of just that, are becoming more common because all the factors that make new infections emerge are amplifying, not retreating. And so we're probably going to see more of this kind of thing going forward into the future. But let's hope this present outbreak is just that, a small outbreak that subsequently now comes under control and we don't see any more repercussions.
Yeah. If the origins are South America, what are the chances of there being maybe more significant infections there?
Well, hantaviruses are all over the world. They're in many, many countries, and there's a family of them.
Yeah.
You find them in many, many places because they're actually carried by wild animals. This is a zoonotic infection. It's not a human infection. And it's only occasionally that it jumps into us as an incidental so-called dead-end host. And that's why actually they have such high fatality rates, because these viruses are completely foreign and unadapted to us, and we're unadapted to them. So we're not good bedfellows, and you tend to get very dramatic consequences. It's a bit like rabies, which will live happily in a bat, but will kill a human very, very quickly. It's very similar. It's that difference between one animal species and its adapted virus and us. The origin is almost certainly South America. We know based on incubation periods and so on, it has to have come from Argentina. And this is also backed up by genetic evidence, because very promptly now you can read the genetic codes of infecting entities. When someone presents with an infection, known or unknown, you read the genetic code of that infection entity and you compare it to a database of what we've already documented from around the world, and that enables you to pinpoint pretty precisely what the cause of the infection is and therefore where it's likely to come from. Many of these infections have restricted geographical ranges. This is no exception. So it points to an Argentinian origin, and one specific lineage of this group of hantaviruses called the Andes or Andean strain, and that is currently circulating in Argentina. So therefore the most likely scenario is that somebody, before they boarded the boat in Argentina, which left on the 1st of April, encountered the virus in the environment. They must have done that because it's carried by wild animals, rats and mice. It's not carried by humans. So they must have brought it aboard, or there were mice and rats on the boat already that gave it to them on the boat. Now, the company have denied that. They say they don't have a rodent problem on the boat, but I haven't seen any evidence either to support or refute that hypothesis. But it's absolutely the case that it must have come from Argentina originally and then got into the people. The open question is it going person to person? These hantaviruses don't commonly do that, although there is some evidence that this particular hantavirus, the Andes hantavirus strain, might show limited human-to-human transmission potential, but it's very inefficient in doing so. We're talking about single numbers of % of cases that can do that. So therefore, we are not. And the WHO in their press conference yesterday were emphatic, saying this is not an epidemic, this is not a pandemic, this is not COVID. I'm quoting Maria van Kerkhove there, who's their Director of Emerging Infection. They were very eager to reassure people that we're not looking at what will become a massive outbreak across the world or anything like that. It's interesting nonetheless, and again reminds us that the threat from emerging infection hasn't gone away, because all the factors that drive new infections to emerge and to spread amongst humans are amplifying, largely because of human population, human movement, human migration, human conflict with nature.
And I think we needed a little bit of that assurance this morning. Thank you, Chris. Perfectly timed. I have been in my season of reconciling time, and I went down that rabbit hole. I know we can measure time to a billionth of a second. We've got atomic clocks that can keep accurate time for billions of years. But I believe we still don't actually know what it is. Is time a physical thing that exists, or is it just our way of, what, bookkeeping?
Well, we know that things happen over time, but we've invented a way of measuring it. But time exists whether we measure it or not. It's like that philosophical argument, if a tree falls over in a forest, does it make a sound if we're not there to hear it? Well, time ticks because we know that chemical reactions happen at a rate, but our way of measuring them, we call it the second. We define that actually in terms of a physical activity. It's how many times a cesium atom vibrates in a cesium clock, billions of times a second. That's our definition of the second. We used to define a second in terms of the length of a day, which was the rotation of a planet, and we worked it out on the basis of how long it took the Earth to go around the sun. Now we do it with a physical property, but time definitely exists in that regard because the universe has been aging, and it was doing that for billions of years before we came along and then started measuring it. So we know that time and the arrow of time exists and exists independent of us measuring it, but the way we define it and choose to measure it, well, that's a human construct.
And time goes both forward and backwards.
Well, we certainly can time travel, but we haven't found how to go backwards yet. Stephen Hawking famously said, I don't think time travel can be possible because otherwise we would have been invaded by tourists from the future. And I'm inclined to agree with him. It's very, very difficult to wind time back. I don't think it would be possible to go backwards, but we can certainly time travel by going forwards, and we can change the rate at which light travels. We can change our perception of time and speed, and that's what relativity is all about. I mean, it's mind-bending stuff, and that was the insight of Einstein 100 years ago. We can certainly distort time, but we can't go backwards in time. You can go faster or slower forwards in time, but I think I'm inclined to agree with Stephen Hawking, inexpert as I am, that we're not going to see us going backwards in time anytime soon.
Okay, let's go to, is it Jason? Jason's got a question.
Hi, good morning, Clarence. This is Jason here from Brackenfell. A quick question for the great doctor. How do trees know when it's autumn and spring? How do they know when to shed or drop the leaves and when to start blooming again?
Hi,
Just
Jason.
curious.
Well,
Thank you.
the answer is that they're very tuned into their environment. Now, of course, for some trees in some parts of the world, it isn't autumn and spring. If you live in the equator, then you have summertime all year round pretty much. And so trees there behave quite differently. But trees are really tuned into their environment, and they know what time of day it is, they know what time of year it is. And in fact, plants can do really quite complicated mathematics because they do all their growing at night time. They grow using energy stored up during the daytime, and they grow at just the right rate so that they will run out of energy just as the sun is coming up, so that they can then begin to rebuild their energy stocks and not dent their growth. And that means they have to keep tabs on time. So they have a day clock, they have a body clock running in them, just like you and me do, but they also have an annual clock. And that annual clock is not set by daylight so much, but it can be set by day length, and it's also very temperature sensitive. So plants have processes which are responding to average daytime temperature. And when you have sustained higher average daytime temperature after a cold snap, the plant can interpret this as days are warming up and lengthening, therefore it must be spring. And when you have the reverse equation, which is days are shortening, lower average temperatures and some real cold nights, well, we must be going into winter then. So that's a cue, I can drop my leaves. And so plants use these cues from the environment to time what they do at different times of the year.
We have in Paarl, not too far away. I think it's Paarl's best kept secret. The Arboretum there, subdivided nearly 3 kilometers of land, subdivided into all the continents with trees from those continents, then represented there. And they've certainly adapted to our conditions in the south. So they will lose their leaves in anticipation of winter. That's just fascinating, isn't it?
Yes, and the other thing is you can jet lag plants. And a friend of mine works on what we will do when we want to grow plants in space, because when we send plants into space, they won't have a 24-hour day. If you take them to Mars, for example, to grow food, if we colonize Mars, well, how are we going to get around that one? So they're looking at the body clocks of plants in order to work out how to compensate for plants being jet lagged just like we do, because they are so tuned into these timings. And yet you have to work out ways to make the plants capable of tolerating those sorts of conditions, either by adjusting the way their body clock works or making their conditions a bit artificial to make sure they don't realise they've moved from Earth to Mars.
Okay, some people are smelling a rat, and it is, of course, related to the hantavirus. The question is, please ask how viruses are isolated, as I hear they use symptoms to identify the hantavirus. And another related question, I'd like to know how it was contracted. Is it by eating a rat?
Right. Well, first of all, how do we identify cases? Well, we have what's called, in any kind of outbreak, a case definition. And in a case definition, you list down the main presenting features, which would be a fever, usually, flu-like symptoms, headache, muscle aches and pains, a cough. So a set of symptoms that we know usually define people with that particular condition. Then we take samples. And there are various samples you can collect from people. They'll be determined by what sort of infection you're dealing with. If it's a respiratory infection, you might want nose, throat swabs, perhaps some sputum from the chest. If it's a systemic infection, you probably want to anyway, but you'll want some blood. How are they tested? Well, in some cases, with certain infections, we grow the agent. If it's a bacterial infection, you can take those samples, sweep out an agar culture plate, and you grow the bacterium, and then you confirm that's what you've got. With some infections, we look in the blood for either the product of the infection, so-called antigens, which can be picked up by serology tests. We can also look at the body's reaction to those antigens by making antibodies. When a person's infected, they often mount an immune response quite promptly, and this will produce certain classes of antibody which we can detect, and that's a serological response again. You can also, especially more recently, look for the genetic material of various infecting entities, and this is a very fast, very agile diagnostic measure because it can pick out tiny amounts of the thing that's the cause of the infection. And because it uses the polymerase chain reaction and amplifies that message, you can pick things up very early from very tiny amounts of material. So this is one of the main kind of heavy lifting machinery now of doing diagnostics. We do these PCR-based tests. So usually all of and some combination of the things I've mentioned will be used in order to work out who's got something, what they've got, and also identify more specifically what that particular infection is. So in the case of the hantavirus, we're doing nose and throat swabs, and we've got a couple of people who are isolating in the UK at the moment, more expected to arrive home. They're isolating themselves at home and keeping an eye on their symptoms. So we're using both clinical reports of how they're doing, but also swabs from nose and throat and also some blood samples. And if they do develop symptoms, we might get some urine as well, because we know that these viruses make a beeline for the kidneys in some cases, and therefore virus components might be shed into the urine, which we can also detect using all of the above tests I mentioned.
And it is.
Oh, there was another question, wasn't there, about eating rats? Right.
Yeah. How do they contact?
The route of and the other question about the route of transmission of hantavirus, the natural host are rodents, mice, or in the case of this Andes strain, the pygmy rat. Those animals carry the virus and they shed it in feces and urine, but it's also in saliva. So there are a range of ways you can contract it. One is by breathing in particles of virus which have been swept up in the air by air currents from dried urine or feces. The other is if there are body fluids like saliva there, or you get a bite from one of these animals, that could potentially and theoretically transmit it. But the most common way that's been documented with people acquiring hantavirus is breathing in particles from dried out urine or from feces that have contaminated an environment.
The Naked Scientist with us, Dr. Chris Smith. And if you have a question, drop it via WhatsApp 072-567-1567. And you can call on 021-446-0567. We'll prioritize your call. Voice note from Mark. Let's take a listen.
Good morning, Clarence. If you could ask your guest, I've got 13 wristwatches, of which I probably use 4 often. And the rest of them have all stopped. And they've all stopped at the exact same time, which is 9:15. I mean, I find that so weird. Is there an explanation for that?
Is that AM or PM? Dr. Chris.
I think someone's playing some tricks on him, don't you? The likelihood of a watch stopping at an identical time, and all of them independently doing that, I think is too much of a coincidence, because, I mean, even though a broken watch is still right twice a day, the likelihood of these other watches all breaking down or running down at exactly the same time seems a bit spooky to me. So I suspect that somebody's been in that watch drawer and they've twiddled that little knob on the side and set the hands all to 9:15 and then said, gosh, this is spooky, isn't it? Too much of a coincidence otherwise.
That's why I asked, is it AM or PM? Because then we know, I know what Mark does at 9:15 PM, if you know what I mean. But let's go to another question. I was always curious as to why there are near no tides in the Mediterranean Sea and big tides in the Strait of Hormuz, even though Hormuz is smaller than the Mediterranean, and they have a similar sized openings to a bigger sea. Interesting.
Okay, well, I suppose upstream, excuse the pun, of this question is, well, why do we have tides at all? And why do we have 2 tides a day? So let's address that first, because that helps to understand what is going on. The Earth has a large moon. We see it in the sky. And the Earth turns, taking 24 hours to do it, inside the orbit of the moon, which takes 1 month to go around the Earth. So we're spinning inside the moon's orbit, and because the moon is really big, it's unusually big because it was made in a slightly unusual way, it pulls the Earth towards the moon. The Earth doesn't move because it's much bigger than the moon, but the water on the surface of the moon, on the surface of the Earth, does move, and it therefore heaps up on the side of the Earth close to the moon, and you get a bulge. You also get another bulge opposite that first bulge on the opposite side of the Earth, because that's farther away from the moon and therefore isn't being pulled as hard as the rest of the water around the Earth's surface. So that's why you get 2 tides a day, because the Earth turns through the first bulge, which is the one closest to the moon, and that's usually the higher of the 2 tides each day. And then when you get round the other side of the Earth, you pass through that second bulge. That's your second high tide. So in any 24-hour period, you get 2 high tides and 2 low tides. Now, because the Earth is turning through those bulges, it's not that the tide is moving round, it's that the planet is moving through the bulge. And this means that you're basically pushing water into all of these bits around the Earth. Wherever there's a landmass, the water is trying to flow over and around those landmasses in order to stay close to the moon. And it gets funneled and pushed around and directed and builds up momentum as it goes. So when you've got places like, if you take the UK, you think about the map of England, and it's got that long bit that stretches out towards America, which is the West Country, and Wales, which is above it. You've got the Bristol Channel and the River Severn between the two. And you get some of the highest tides in the world there, maybe 10, 15 metres when you have an exceptional tide with a spring tide, for example. Why should that be so massive? Well, the tide has come across the Atlantic, uninterrupted, building up enormous momentum from America, and it then gets thrown up the Bristol Channel and funneled in because it's being collected across a large area and pushed up that waterway, and you get this enormously high tide. The Mediterranean, on the other hand, has a relatively restricted inlet to the Med between the top of Africa and the south part of Europe. So it's hard for much water to get in there uninterrupted. And then the tidal effect in the Mediterranean itself, because it's a relatively small body of water just across the top of Africa, bottom of Europe, you don't have that same momentum being built up. So you don't get that huge tidal range in the Med like you would in the west coast of the UK, for example, where it's got the whole of the Atlantic to build up momentum uninterrupted and then be funneled up the side of our country. So that is why you get high tides in some places and less high tides in others. So where you've got areas where you can direct water, it can build up a lot of momentum and then flow through a narrow area, lots of water going through a narrow area. You build up a large body of water because a little goes a long way. When you've got not much water entering a relatively big area of the Mediterranean and then not much opportunity to build up more momentum and movement across that body of water, you get lower tides.
We got Jen on the line from Constantia. Welcome, Jen. Go ahead for the Naked Scientist.
About hantaviruses and something just clicked. There's a project that they've trained giant cane rats to identify or to diagnose TB by sniffing different samples of sputum. So my question is, are the rats at risk of actually contracting TB or some form of TB? I mean, obviously I know TB human, not rat, but things can go from through different species. Is there a risk that by sniffing lots and lots and lots of samples, positive samples of TB sputum, that the rats could in fact be infected with TB or whatever they happen to be sniffing?
The line isn't great, Jen, I'm afraid, but I think what you're asking is if you get animals sniffing things, can they catch those things? And the answer is that with mycobacteria, which TB is one example of a mycobacterium, these are very infectious and pretty much all animals can pick them up. And certainly they do spread through the air. So if you have infectious samples of TB, some of the microbes, that it's a bacterial infection, can become airborne, aerosolized, and if you breathe them in, then they can settle in the airways and they can establish new infections. So with TB, for example, one of the most common ways it spreads is that a person with TB coughs because it causes inflammation in the lungs. The lungs respond to inflammation by coughing, and this expels from the infected person a mist of droplets, and in those droplets are TB bacteria. And if you then breathe those airborne droplets in, they settle in your airways and they can establish new infections. So anything that aerosolizes the bacterium makes it potentially able to be breathed in by whoever's doing the breathing, and it could then establish infection in that individual. Now, different mycobacteria are more virulent than others, and so there are lots of mycobacteria that live in the environment which are, with a person with a healthy immune system, totally harmless. But in a person with HIV, they can be deadly. And there are similarly some mycobacteria that are very infective for certain organisms, but totally harmless to others. And so human TB is very highly optimized to spread among humans, but can also affect other animals. Bovine TB is very good at spreading in cows and getting into other animals like badgers, but can also occasionally jump into humans. So it's horses for courses. Different animals have different susceptibilities, but the general rule of thumb is if you have something that aerosolizes the infection, it can be breathed in by a third party and set up a new infection.
Maybe two quick little questions I can throw at you from Didi. Do meerkats also carry the hantavirus? And from Fatima, does cannibalism cause a disease of the brain?
Meerkats, I don't think, are hosts of hantaviruses. I think it's specifically rats and mice. But if anyone knows different, do please tell me. Cannibalism definitely does cause brain diseases. If you are a cow eating more cows, you can get BSE, bovine spongiform encephalopathy, which is exactly how the mad cow epidemic broke out in the UK, with people rendering cows and feeding neurological tissue back to cows. And it led to the concentration of a prion disease, BSE in that population and subsequently exposed humans, and there were human cases of mad cow disease. In humans, there is a similar condition which is called, what's it called? It's in the Four people in Papua New Guinea. Kuru, is it? And these people ritualistically were eating the brains of their relatives, and this was producing this condition in these people. You would see it was mainly the women that were doing the consumption, and they were manifesting these degenerative states, and subsequently it turned out that they were developing the human equivalent of mad cow disease, because again these prions, which are infectious proteins that build up in the nervous system, can transmit from one person to another if you are exposed to neurological tissue containing a high burden of those prions. And so when the people were cannibalizing their relatives, then they were exposing themselves. This was leading to the infection in them, and then when other people were eating them, there was already a higher infectious burden, so it increased the rate at which it transmitted, and so it went round like a vicious cycle. And when they stopped doing the cannibalism, the disease went away.
Fascinating, as always. And a big thank you to consultant virologist at Addenbrooke's Hospital in Cambridge, Dr. Chris Smith, the Naked Scientist, every Friday, just after 9:30, live from London today, with a time of 2 minutes to 10 o'clock. Ole Kalinga standing by with Eyewitness News at the top of the hour.